Today we are going to talk about point mutations. Point mutations are single nucleotide changes in DNA. There are two kinds of point mutations, transversions and transitions. Transversions are changes between different types of bases, and transitions are changes between bases of the same type. There are four different effects of point mutations, and we will talk about them all. So here's the normal coding strand of DNA, and below it I have written the mRNA strand, so the thymine is exchanged for uracil. The next thing to do is to translate the mRNA and write out the amino acid sequence. So AUG is the start codon and codes for methionine, UGG codes for tryptophan, AAC codes for asparagine, GCU codes for alanine, UGC is cysteine, and UGA is the stop codon. Just for our knowledge, CCC codes for proline and GGC codes for glycine. So say the point mutation is a transversion at this third codon, and the cytosine is replaced with an adenosine. Then this would be the mRNA transcribed, and during translation, the start codon would be the same, so would the UGG coding for tryptophan. But whereas the original amino acid sequence had asparagine next, AAA codes for lysine. The rest of the sequence is unchanged. This single nucleotide substitution changes an amino acid, and we call this a missense. The amino acid change could affect the protein function, and if it does not, it is neutral. Now say that there was a transition at the same location and cytosine was substituted for a thymine. Then this would be the mRNA. The start codon methionine would be the same. So would the tryptophan. AAU codes for asparagine, which is the same amino acid as in the original sequence. So the protein is unchanged, and we call this a silent mutation. Now let's model a transition point mutation at the third base of the second codon, changing the guanine to an adenosine. Here's the mRNA with uracil in place of thymine. AUG is the same, still the start codon. UGA is a stop codon, so here the translation stops. When the mutation results in an early stop codon like it has here, we call it nonsense. To model the fourth and last effect of a point mutation, let's substitute this guanine in the sixth codon for a cytosine, which is a transversion. Here's the transcribed mRNA. Now the first 5 codons are unchanged, so they code for the same amino acids. The sixth codon is UCA, which codes for serine, then CCC codes for proline, and GGC codes for glycine. And translation continues. In the normal amino acid sequence, we had a stop codon after cysteine. The point mutation changed the stop codon to a serine codon. When the stop codon is changed into a non-stop codon, we call it a read-through.